Okay, in chapter 3, we're going to talk about one branch of chemistry that's especially relevant to biology, and that's going to be organic chemistry. So, organic molecules are really the backbone of living things. These molecules contain both carbon and hydrogen atoms, which sets them apart from inorganic molecules like water and salt, which might contain carbon or hydrogen, but not both in combination. So, what makes carbon so special? So, for starters, it has 6 electrons total, 4 in its outermost shell or its valence shell, meaning that it needs 4 more to become stable to fulfill that octet rule. So, this makes carbon incredibly versatile because it can form 4 covalent bonds with other atoms, which is going to allow it to create really complex structures that can be chains, rings, or even branches. So carbon's often going to bond with essential elements like hydrogen, oxygen, and nitrogen that we talked about previously that make up 96% of the mass of all living organisms, and it can even bond to other carbon atoms. So when carbon bonds only with hydrogen, we get something called a hydrocarbon, which is going to form the foundation of many biological molecules, including fuels. So even more fascinating is that carbon compounds can exist as isomers, so molecules with the same number and types of atoms, but they're going to be arranged differently. And these structural differences can lead to huge differences in properties, even if the molecular formula is the same. So carbon atoms form the skeletons or the backbones of organic molecules. And these skeletons can vary both in size and shape, so they can be straight, branched, or ringed. But the real chemical personality of organic molecules is going to come from its functional groups. So these are specific groupings of atoms that behave in a predictable way. And functional groups are like chemical add-ons. So no matter which molecule they're attached to, they're going to react in the same way. So for example, if we have a hydroxyl group, which we talked about previously, which is the OH group, will always make a molecule more polar and likely to dissolve in water. So in molecular diagrams, you'll often see a generic R symbol. So we can see a couple over here. And that's going to stand in for the rest of the molecule, while the functional group is really going to be the star of the show. So all living things are going to be built from 4 major categories of biological macromolecules. And our 4 macromolecules are going to be our carbohydrates, our lipids, our proteins, and our nucleic acids. And these are often called macromolecules because they're made up of many smaller subunits. So during digestion and metabolism, our body is going to break these down into their building blocks, which we refer to as monomers, which are then going to be reassembled into molecules that our cells need in order to grow and repair and to function. So think of monomers as individual like Lego bricks. When you snap them together, you get a polymer, which is just a long chain of repeating units. And this process of linking monomers together is called a dehydration synthesis reaction, because it's going to involve removing a water molecule in order to link the monomers together and form a new bond. So in order to break these bonds, we have to do the reverse process, which is a hydrolysis reaction. So we have to add water back to basically a hydroxyl group to one monomer and a hydrogen to the other monomer. And this is how our digestive system is going to take complex molecules like starch or protein and break it down into usable glucose or amino acids. So that's what it's showing you over to the right in the image. In order to link monomers together, we remove what's in red here, so a hydroxyl group and an oxygen. So we're essentially removing water, H2O, so that's why it's called a dehydration reaction. In order to take our macromolecule or our polymer and break it apart, we have to do the reverse process, which is add water back. to get individual monomers. So it doesn't matter which macromolecule we're talking about, if it's nucleic acids, proteins, lipids, or carbohydrates, we, in order to make these macromolecules, have to undergo dehydration. In order to break them apart into their monomers or their building blocks, they have to undergo hydrolysis. It's going to be the same reactions for any of our 4 macromolecules that we have.